of a change of pace in a certain sense. In organizing this particular symposium, it seemed important to reach to future technology, and particularly that which has to do with biocatalysis. Frankly, we found frequently that people were very enthusiastic about biocatalysis but had little understanding of really what the situation was. We have now to hear from Professor Frei prospects for improvement in biocatalysis for large-scale applications. By the way, I invite some of the people who are toward the back to come forward. Please, good seats up forward.
Thank you, Alex. It gives me a pleasure to address this meeting. Thank you for coming. I'm here to tell you something about biocatalysts, and my specialty is enzymes. I'm engaged in basic research on the mechanism of action of enzymes. And I have of course, high regard for enzymes as catalysts, but essentially as biocatalysts in the living organisms. They are, of course, the catalysts of life that catalyze our own bodily processes. They're very highly efficient and are attractive potentially as industrial catalysts, but they haven't come very far. I'd like to start with the first slide. May I have the lights down? There's some fairly familiar information about enzymes. First of all, enzymes, of course, are proteins. Very often they have collaborating molecules, coenzymes, or other cofactors that are required in catalysis to make a complete enzyme. But basically, they are proteins, which are polyamino acids, whose structures, whose three-dimensional structures in solution are really determined by secondary valence forces rather than by covalent bonds alone. And that presents the enzymes with certain capabilities that are not available from any other catalysts, but also with certain limitations that are not faced by other catalysts. And I'd like to emphasize both sides of that story during my talk today. The advantages are listed here, the potential advantages on the left side. They have very high catalytic rates. The enzymes typically accelerate rates by factors of 10 to the 8 to 10 to the 12. And I should say that this is a range. We know both lower rates and higher rates. The lower rates normally refer to reactions that have spontaneous rates that are relatively high, and the enzymes are pushing the fusional limits, and so are only 100 to 1,000,000 times faster than the spontaneous rates. And then, of course, there are enzymes that catalyze reactions at with rate acceleration factors greater than 10 to the 12, whose rate acceleration factors often cannot be accurately measured because the spontaneous reactions are so slow. But we would know there are cases of 10 to the 14th and probably higher for rate acceleration factors by enzymes. Another important advantage of enzymes is that they have a very high reaction selectivity. A given enzyme tends to catalyze a particular reaction and not other reactions. Perhaps the most useful aspect of enzymes as catalysts is their stereoselectivity. They are almost invariably, I should say almost, because I happen to have a research interest in enzymes that are not stereoselective, because I don't understand how it works. But they are generally very stereoselective, and that is often useful. They work under very mild conditions, typically 25 degrees, pH7, in aqueous solution. We get these very fast rates. And they generate very few byproducts because of the reaction selectivity exhibited by the enzyme. So product workup should be relatively simple. Now, on the other side, the limitations. For the use of enzymes, a principal limitation is expense. I gained a very high regard for this limitation the first time I purified an enzyme. Purification of enzymes tends to be labor-intensive, but there have been many advances in the technology of enzyme purification over the past 10 years, It is not nearly so labor-intensive as it was, but I will not discuss that today. It basically involves fairly familiar kinds of techniques such as affinity chromatography and so forth. I'll discuss, however, some new methodologies, some new genetic methodologies that enable one to produce enzymes in larger quantities and higher concentrations, and thereby reduce the purification problem and the expense of enzymes. Then another very important problem with enzymes is their lability. As I indicated in defining for you what enzymes are, their structures, three-dimensional structures in solution, are determined by secondary forces, hydrogen bonding, electrostatic interactions, the hydrophobic interaction, London forces, which are individually relatively weak. And this tends to cause enzymes to have low thermal stability. So that's a real limitation for enzymes, thermal lability. There are other problems of lability. They're often poisoned by heavy metals or undergo oxidation reactions readily that cause problems with stability. So reusability becomes a problem with enzymes due to this lability problem. Now, another limitation, only in a certain sense, is substrate selectivity. Oftentimes, for specific processes, a specific substrate needs to be processed, and that substrate selectivity is then not a disadvantage. But often, for more general purposes, enzymes for general use, because of their high degree of substrate selectivity, can often be used for a wide range of substrates very efficiently. It would be good to be able to control that in some way. And I will spend a little time today discussing the first initial efforts that are still in progress or are being initiated to alter in predictive ways the substrate selectivities of enzymes. And then there is the aspect of product inhibition. Product inhibition is a problem for a certain percentage of enzymes that limits their applicability for batch processes. Enzymes in their natural milieu are usually not limited by product inhibition because in the living cell, their product is being consumed by the next enzyme in the metabolic pathway, so products are being continuously removed. About the only way to handle the product inhibition problem when it occurs, and it doesn't always occur in a batch process, is to find a means of removing the product. When I've listed here a few of the industrial applications, and I will extend this list in a particular way. The industrial applications that are already, that enzymes are already used for are sugar transformations, the glucose isomerase system, semi-synthesis of antibiotics, steroid transformations, synthesis of proteins, synthesis of amino acids, synthesis of amines, and synthesis of nucleotides. Now, I just have a few comments to make about this list. First of all, the list is quite short. It tends to involve fairly specialized molecules that are not produced in huge quantities industrially. It tends to involve complex molecules with a large number of stereocenters, as in antibiotics, sugars, steroids, proteins, amino acids, all of these nucleotides. All of these include molecules of some complexity with regard to stereochemistry. And that really, I think, illustrates, as clearly as can be illustrated, the principal advantage of enzymes is in the stereospecificity of their reactions. Now, this list, in terms of total industrial applications, would be extended by two things, which I will mention. But before I do, I'll just point out that this particular list is one that is devoted to synthesis of products. There are other industrial applications of enzymes where enzymes really are quite important and will continue to be important and have growing importance. One of them is well known as product additives to detergents to aid in the dissolution of soils of natural origin. And the other very important and big growth area is in the diagnostics field. Enzymes are very, very carefully researched for use in medical diagnostics, and that's a very rapidly growing field. The industries are after my graduate students en masse, but those are not what one would consider to be large-scale applications, which is what I have devoted my comments primarily to today. In the Medical diagnostics field, the problem of labelability of enzymes is not a very serious problem because the enzymes are used once only for a diagnostic test and then discarded, and they're not recycled, so many of the problems that exist for large-scale applications do not exist in the applications of medical diagnostics. Now I'd like to turn then to, backing off here to my preceding slide, methods that are being researched and for which there is basic knowledge available to reduce the expense and to control the labelability and substrate selectivity of enzymes. These methods have come from the biological side rather than the physical side of biochemistry. They're basically genetic methods, and they consist of the following. Each enzyme has its structure specified by the structure of a gene in the DNA of a living cell. And today now, using modern molecular biological techniques, it is possible to isolate the gene for a specific enzyme. That gene has a nucleotide sequence that specifies the amino acid sequence for an enzyme or other protein. And the genes can be isolated by a variety of techniques. We can't go through all of these techniques. Some of them involve systematic fragmentations of the DNA and then derivatization biologically or biochemically of the fragments, screening for and isolating the specific fragment desired. Other methods are also available, but I won't go into them. The point here that I want to emphasize is that it is possible for enzymes to have the gene, and then it is possible to obtain the nucleotide sequence of that gene. That is a relatively straightforward affair, and it is routine then to translate that nucleotide sequence into the amino acid sequence for the protein, in this case enzyme. Once the gene has been isolated, it can be inserted into a high expression vector. The high expression vector here is a fragment of DNA, an extra chromosomal element that may be found in a microorganism, and that is part of the normal genetic complement of that organism, but can be obtained, opened up in specific ways, and can be used to trap the gene within its own DNA, so that the DNA, the foreign DNA from the isolated gene, can be inserted into an expression vector. That expression vector, which is a small fragment of DNA then, can be reintroduced into the microbe and will replicate itself many times. Of course, each time it replicates itself, it replicates the desired gene. And therefore, that organism will have a higher than normal complement of that gene, of the desired gene for that protein. And this is one of the major factors, one of the major means by which the expression of a particular enzyme in a microbe can be amplified by the high expression vector. There are other methods of high expression or other mechanisms of high expression than simply the replication of the vector, but that is one of them. Well, when the microbial culture has been transformed with the vector, individual microbes containing the vector with the foreign gene can be isolated by screening techniques, and that's called cloning, and then an individual microbe with the appropriate vector and gene can be cultured, reproduced, and grown up in large cultures. So by that technique, it is possible to overproduce the enzyme. The overproduction is due to the high expression vector from the gene. Now this gene might be a human gene, or it might be some animal gene that has been then cloned into a microorganism that can be cultured. But now by this technique alone, and we can't go into all of the vagaries of this, it is possible to produce enzymes in very much larger amounts than has been possible in the past. Now I just want to mention one other thing. Once that gene has been cloned into an organism, isolated and cloned, the nucleotide sequence is known. Methodologies are now available for altering the nucleotide sequence at specific sites. This enables one, by altering the nucleotide sequence then, to reclone the mutant gene, express the mutant in culture, and isolate the mutant protein. The protein mutation has resulted from the mutation in the nucleotide sequence. So that it is possible now to alter the structures of enzymes and to do structure-function analysis on purified enzymes with defined structural alterations. I emphasize that the change in the structure of the protein here is really in the amino acid sequence, and we do not know the translation rules for translating amino acid sequences into three-dimensional structures yet. So there is a considerable element of chance involved in experiments of this type. Now I want to talk a little bit about solving the lability problem. There is really no solution to the lability problem. There are ways of attenuating it and controlling it. As I indicated, the first time I purified an enzyme, I gained the appreciation for how delicate they truly are in structure. One has to be rather careful in dealing with it. However, it is also a fact that there are microbes called thermophiles that grow at very high temperatures, even at temperatures higher than the boiling temperature of water. And the enzymes inside these organisms must be surviving that heat. So clearly, there are ways of stabilizing proteins to thermal conditions. Those organisms seem to have solved that problem, but I would like to emphasize that one of the major means for stabilizing enzymes to heat is immobilization on solid supports. I won't discuss it further. It's an area of very active investigation over the last 15 years or so, and methodologies for immobilizing enzymes are well advanced in development and continuing to be developed. I will emphasize, however, that there is one way in particular of immobilizing enzymes for use that is easy and cheap, and that is to leave the enzyme inside the microbe and simply use the microbial culture as the catalyst. So that in those cases, then one doesn't have to bother with the purification. One can simply use an overproducing strain of a microbe growing in culture to catalyze the transformation that we might desire. Some of this has been done industrially, and I predict that there will be much more of that done industrially in the future. Well, how about other means of stabilization? I'm just going to show you two. One of them has been realized. This little drawing simply represents the chain, the amino acid polypeptide chain of a hypothetical protein. We're not showing very much of the structure except the amino terminal end, the carboxyl terminal end, and we're indicating here that there is a definite three-dimensional arrangement or conformation that is governed by the secondary valence forces. Now, if that structure is the structure that is desired, we know that what happens with heat is that the vibrations of the molecule begin to take over and overcome the structure, and the structure tends to open up. If we look at this structure, we see it consists of two domains, a folded structure on the left and a structure on the right. One of the things that can be done and has been done is to insert disulfide bridges between crucial structural elements to hold them together. Then the structure is stabilized by covalent bonding rather than exclusively by secondary valence forces. And this has actually been done. In the favorable cases where it has been done, there has been a cysteine already present in a logical site. And what has been done is a cysteine has been introduced in a companion site nearby from another segment of the chain. And then the daughter molecule has undergone spontaneous oxidation to introduce a disulfide. And that has led to very significant increase in thermal stability. And I think that this kind of an approach can be used for aiding in the stabilization of enzymes by genetic engineering to enhance their thermal stability. Now then, there's another thing that has occurred to me that, so far as I know, has really not been an active area of investigation. Many enzymes live in a reducing environment in cells and have these cysteine SH sulfonyl groups on the surface. Now these groups, unless they play a crucial role in catalysis, can cause a lot of trouble and really be nothing but trouble for a purified enzyme living in, say, an oxygen atmosphere or an air atmosphere. For one thing, they're very highly susceptible to oxidation, can form disulfide bridges both intramolecularly and thereby distort the structure, or intermolecularly and precipitate the protein. Furthermore, they're subject to coordination with heavy metals, and this is part of the reason for poisoning by heavy metals. But when those SH groups are actually not involved in catalysis, it very often does not make very much difference whether they're there. And it is possible by directed mutagenesis to change them from cysteine to serine, which has OH groups, and don't cause such problems. And this has been done in a few cases, not for the purpose of relieving poisoning by metals, but just for examining whether the SH is crucial or essential. It turns out that very often it is not. And I would think that an enzyme whose use in industrial application is contemplated, the investigators would study the SH groups and see which ones are needed, which ones are not, and those that are not would be excised. Now I'd like to go into some chemistry for a few minutes and discuss two aspects of the engineering of biocatalysis. This is not my field, and I have chosen the work of two investigators from actually California. Jack Kirsch and his associates at UC Berkeley have studied aspartate aminotransferase. Aspartate aminotransferase catalyzes an overall reaction, and this reaction shown is happening. The structure shown here is pyridoxal phosphate. That is the coenzyme derived from vitamin B6. It is the coenzyme that's involved in transaminase reactions, aminotransferase reactions, and this enzyme catalyzes the reaction of the aldehyde group of pyridoxal phosphate with the amino group of amino acid, in this case aspartate, carboxymethyl for R, to convert the amino group of the aspartate to a ketone, making oxaloacetate, and the aldehyde group of pyridoxal into a primary amine. This is pyridoxamine phosphate, another form of vitamin B6. This is really the first step of the transaminase reaction. In the second step, a different keto acid reacts with the pyridoxamine, and the reaction in the reverse direction results in the transamination process. The structure of this enzyme in 3 dimensions is known for protein X-ray crystallography. And the binding site for aspartate has been defined. The aspartate with the carboxymethyl R group has a negative charge on the carboxyl group, and the binding site in this aminotransferase has in the binding cleft an arginine residue, which is a basic amino acid positively charged, pK 12.5. 3 methylene groups here, deep in that binding cleft. Now the enzyme for the reaction shown has the following kinetic properties for arginine itself and for aspartate. Remember, arginine is an amino acid too. Kcat over Km is the second order rate constant for this reaction. These are just the Michaelis-Menten saturation kinetic parameters. Is 1.9 times 10 to the 4th per molar per second. There should be a per second there. That was cut off in making the slide, apparently. For aspartate, for arginine, it's about 10 to the 6th slower, 2.8 times 10 to the minus 2. That's the native enzyme. Now what Jack Kirsch and his associates did is he reversed the polarity of this reaction. He took arginine out of position 292 in the enzyme and replaced it with aspartate. And then he tested that mutant enzyme, arginine 292, as an enzyme for transamination against aspartate and arginine. And what he found, you see now that is a complementary change. The same number of atoms and groups are involved in the bound complex and in the same apparent geometry, unless the enzyme structure itself changes a great deal. And the experiment was designed to find out whether the enzyme specific for a negatively charged substrate could be made specific for a positively charged substrate. The reaction, the experiment worked. The Kcat over Km for the mutant enzyme, using aspartate, had a value of 0.06 per molar per second, whereas for arginine, the Kcat over Km was 0.43. Now the magnitude of difference was not as large, and the activity of the enzyme was quite low, but the polarity had actually been changed. That is, the rational alteration in catalytic properties had worked, although the enzyme is still not a very highly efficient catalyst. There would be very good reason to believe that its efficiency could be improved by further mutational alteration, but that has not yet been done. That is in process. Now, the other major chemical approach, well, it's really a biological approach now, is one that has been followed by Richard Lerner at La Jolla and by Peter Schultz at UC Berkeley, and I'm going to just show you briefly a recent communication from the Schultz laboratory. And this has to do with the production of catalytic antibodies, that is, new enzymes. There may be reactions that, for which there are no enzymes, although enzymes are available for catalyzing a very wide range of reactions, including cleavage of unactivated carbon-hydrogen bonds. But in such cases, one might want to have a biocatalyst that could be produced to catalyze such a reaction. Well, there's a great deal known about carbonate ester hydrolysis in physical organic chemistry, and the mechanism has been fairly well worked out. It involves the formation of a tetrahedral addition complex resulting from the addition of hydroxide ion, and the transition state for the formation of that tetrahedral addition compound resembles the tetrahedral addition compound structure itself. However, there are no enzymes for this that I know of. Although there may be some enzymes that will catalyze it, this reaction is not something for which an enzyme has evolved. Now, one of the theories of enzyme action is that enzymes catalyze reactions by binding transition states tightly. A kind of biological molecule that binds molecules tightly is the antibody. Antibodies can be generated against proteins injected into animals. They can even be generated against action is that enzymes catalyze reactions by binding transition states tightly. A kind of biological molecule that binds molecules tightly is the antibody. Antibodies can be generated against proteins injected into animals. They can even be generated against small molecules when the small molecules are attached to proteins. And the cells that generate the antibodies can be isolated and they can be cultured. We won't go through the details of the technology of that. But I will say that the approach that Lerner and Schultz have adopted is to produce stable molecules whose structures are thought to resemble the transition states for reactions that they would like to catalyze. And in this case, they have chosen for their reactions the phosphonate monoesters. Phosphonate monoesters were chosen because they're metabolically stable molecules. The phosphorus is a tetrahedral atom. And in this case, since hydroxide is added to the carbonate to form a negatively charged intermediate and transition state, the phosphonate negative charge will play a role here. The Schultz group actually isolated a monoclonal antibody directed specifically toward this that catalyzes. Notice we have a tetrahedral phosphorus here, but an sp2 carbon there. This monoclonal antibody catalyzes the hydrolysis reaction shown here with a rate acceleration factor of 850. It exhibits all of the properties that we have come to know for enzymes. It is not an extremely efficient enzyme. The turnover number is about 4 per minute. But still, it's an effective catalyst and represents a good beginning of a way to designing new catalysts. Well, our time is running short, so I'm going to go to my conclusions here. I'm afraid I lost a little time at the start with the earlier speaker. But I would like to go to the conclusions because I've said the most important things I have to say now. Costs. Basic knowledge has been extended to isolation of genes and overexpression of enzymes in microorganisms. This automatically reduces costs. Lability. Site-directed mutations of cloned genes can improve stability. But in addition, the overproducing organisms themselves can serve as catalysts. Substrate selectivity. Site-directed mutations can alter selectivity. The rules for this are not well worked out. It is still a basic research area, just as the control of thermal stability is a basic research area. Another basic research area is catalytic antibodies. They are accessible in special cases. They are limited to one-step reactions because the multi-step enzymatic reactions are able to proceed because of the ability of enzymes to undergo conformational alterations in structure that give them the capability to stabilize more than one transition state. The catalytic antibodies can stabilize only one transition state, and so far a method for controlling their conformational transitions is not available. So I believe that we can conclude that, lights up please, enzymes as biocatalysts for large-scale industrial applications are not around the corner and will require considerable research. But progress has been made and considerable progress can be expected.